This is chapter 2, and this one is a little bit of history and a little bit more introduction too before we really get into the specific chapters. Chapter 3 will be a little bit of chemistry. So this is still some introduction, taking things a little slower. All right, so we're going to talk about the history of microbiology. And after we are done with this chapter, these are the topics that you should be able to master, hopefully. Comparing the contributions to microbiology that were made by four of the big names in the beginning of microbiology's life. Write correctly an organism's scientific name. So we'll talk about that. Explain the differences between a light microscope and the electron microscope. We won't focus as much on the difference between transmission and scanning, but that will be part of it. And then identify the general characteristics of the microbes and viruses again, focusing on that prokaryote versus eukaryote, right? We've mentioned that twice already, and we'll mention it so many times again. And then describe the tree of life and its significance in microbiology, because there's so many microbes. So that's what this chapter is all about. All right, so we started out, this chapter kind of opens up with talking about how important Linnaeus was to early science in general, because he really started cataloging all sorts of life forms. And so, new organisms, especially plants, just were being brought to him like crazy. And he's trying to figure out, like, what type of creature is this even? Because they didn't even have that information then. So that was his task. And he was doing that, you'll see, in the 1700s, but even before that, we kind of had to back up and talk about what was happening in the 1600s. But Linnaeus is kind of going to be such a big name on the stage that I think that's why he gets the chapter opener. All right, so the first guy that we're going to talk about is Robert Hooke. And he was starting to look at the tiniest forms of life under just, excuse me, a very, very basic microscope. And he, you could almost just call it a lens, but because it's got more than one lens, that's kind of part of what makes it a microscope. And the fact that it holds still, it's not just a magnifying glass, it's a little bit more specialized than that. But so Robert Hooke, this was, in the late 1600s when he was doing his first work. And so he published this book called Micrographia. So, the small stuff is basically what it was all about. But he was not really looking at moving stuff. So he kind of described some very simplistic things like cork and, found there are some interesting things going on that our naked eyes couldn't see. So we had to have these microscopes for. But then one of my favorites, Antony van Leeuwenhoek, he developed a more powerful microscope, so to speak. This one was so good because it could hold the specimens really still and yet get them close to the lens. And what's kind of cool about this one is that it could focus, it could move the specimen up and down next to this little lens that was right here. And it could also move things farther away and closer to the lens. So now we can see things much more in focus than we could before. Like a magnifying glass has a certain distance away from the object where things look most clear. Well, that's how this works as well. Okay, so what Leeuwenhoek was able to see is moving things, and so that was something that Hooke didn't focus on as much. But Leeuwenhoek said there's little things and they're in here and they're moving, and we can't ordinarily see them. And so he saw, most likely some of the protists, some of those little creatures that kind of look like just miniature bugs. That's kind of what a lot of those look like. And even things as small as bacteria. He could see some big bacteria there, pretty sure. And so he called those things in general. He didn't know what any of them were. He just called them generally animalcules, which is a really fun name because it's part animal and part molecule. Like they're as small as molecules. They're tiny little things. But they must be some type of animal because look at them, they're moving. What else can do that? Can you imagine being Leeuwenhoek and seeing the first microscopic things moving, realizing that you were looking at living creatures of some sort or another, and that it would be something new to science that you had seen and nobody else had ever seen, and you've got to try to figure out what the heck it is you're looking at. And then you've got to start sharing that information. And so thankfully, that information did spread pretty quickly. So Leeuwenhoek and Hook were both working at about the same time. And, you know, these were kind of like just normal guys. Like Leeuwenhoek was just a cloth merchant who made a microscope so he could look at to see if the cloth was really good quality or not. Like, am I actually buying silk, or is there something else in it? How tight is the weave? You know, and instead this is just like his side hobby. And then he ends up being one of the 4 most important people in early microbiology because of that. So those 2 were really on the forefront of just discovering what microscopic stuff was out there, that it was even there. I mean, that was all so new. And so their publications just spread like wildfire from there. And so more and more people saw, oh yeah, you're right, there really is stuff growing in there. Okay, the next one then is Pasteur. So this was definitely later, not quite 100 years later, if you're looking at pure overlapping. But Pasteur was really looking at using the scientific method, like not just, hey, let's look at what's in here and see what's there, but let's figure out some of what it does. Let's figure out what it needs to survive or doesn't need to survive. And so Pasteur came up with this idea that wine grape juice becomes wine because of the microbes that are in there. And before that, they had no idea, which just like blows my mind. How could they not realize that something was causing these things to what we now know as ferment, right? They knew that they took grape juice, they knew they smashed it up, they knew they let it sit, and they knew they got wine. Okay, one of the earliest things that was fermented was mead, which is a honey wine. And so they hypothesized that the first time that they saw or that they found this mead, it may have been natural, like some honey. Hey, lo and behold, it tastes really good and it doesn't hurt you. Um, but doesn't it just seem crazy that they had no idea? I mean, they were making these beverages for so many years, and you know, even in the Viking times, that and before, people knew that fermented beverages were safe to drink, but they didn't know what caused the process. They did not know about the microbes that make those processes work. Okay, so Pasteur was like, there's got to be something to this. And so when he used those little microscopes, he was able to see that yes, there really are yeast cells in here. Now let's figure out what this does. And so he figured out through his process of what we now call pasteurization that that the microorganisms really do exist in there, and that we can cause fermentation to occur or not. And that was the really important thing. So he had his little swan neck flasks that he used, and those swan neck flasks, you know, I thought I had a picture in here. That's why I went to the next slide. I don't see it. So I'm just going to pull up one of those swan neck flasks. Yeah, we'll use this one. All right, so just for the ease of this, I'm just going to share my screen so you can see that flask. Okay, so then I'm going to go. There we go. Sorry, I had to select it. And we're going to go here. Okay, so you should be able to see that now. So these swan neck flasks is what we're looking at here. So this is Pasteur, this is his flask. He would boil them, like boil a grape juice, and then he knew that when he did that he couldn't see the yeast cells anymore. He knew that killed the yeast cells. And what everybody previously thought was that if they thought that the little animalcules would be able to just self-generate, like all of a sudden, boom, there they are. Okay, they had no idea that they could go through the air or that they could exist and not exist. They just thought that they could spontaneously generate, which is what that's called. So anyway, so he boiled the grape juice in one of these swan neck flasks, and in one of them, he would put a little bit of water like this, down in that curve, because he knew then, or hypothesized, that the bacteria or whatever these animalcules were wouldn't be able to go past that little bit of liquid there. Okay, so he did that in one, and then nothing would happen because no thing could get in there after he had boiled it and killed it. And then in the other one, he would do that same thing for a little while, but then after a bit, he would cut the swan neck flask off, and then it would ferment. Okay, and he could show, yes, there's yeast cells in there now, or bacteria, and it would continue to ferment as he expected it to. Go back to this. Okay, sorry, I had to get distracted for a second. There we go. All right, so Pasteur came up with this process of, hey, when you heat stuff up, it will kill the whatever's in there. In his case, yeast in wine. And the result of that was that we now knew that there were microbes that could do interesting things. So where, Hooke was really good at looking at the first still things like cork and seeing that there were tiny things in there. And then Leeuwenhoek was looking at living things. And then Pasteur was really good at saying, oh, well, we can see that they actually do chemical processes like ferment wine or maybe even cause disease. He kind of was the first one who said, I think that these things can cause disease. But he never really pulled together his proof that showed for sure that some of these microbes could cause disease. Okay, so that kind of summarizes that trend so far. Hooke with his still lifes, Leeuwenhoek with his movement, and he names them animalcules. Pasteur being able to say that I can show you that they do cause these chemical reactions, turning grape juice into wine. Okay, so then that led to the germ theory of disease, which is what is Robert Koch's claim to fame. So Robert Koch was the first one that said, yes, microbes do cause disease, and I have now proved it. So what he did was he took an animal that had anthrax, okay, which is found normally in the soil. It's in the soil in Kentucky as well. Anthrax is a really nasty thing. And if you get a very deep cut, it can get into you that way, okay? That's usually how it would get into one of us. It's very rare for us to get it in naturally because our body should be able to fight it off just fine. But anyway, so he found an animal that had become sick and then died with anthrax. And he knew that it had these weird rod-shaped bacteria. He didn't know they were bacteria, but these weird rod-shaped things. And so he could see that in the blood of these animals. And that's what's shown on the top right hand of this picture. That's what it looks like under a microscope. And so he took that blood and purified those rods out of it, okay, so that he only had those growing in there. And then he gave that to an otherwise healthy organism, and that organism died. And he looked at its blood, and he found that, in fact, those rods that turn out to be anthrax bacteria continued to multiply in that new organism. So essentially, he took the disease out of one organism, put it into another, and it made the new one sick. And of course, we know that this happens now. Of course we do. We know that things can be transferred through blood. We know that things can be transferred through respiratory droplets. We know this. But in the past, they did not. They had no idea exactly how all these things moved around. What caused the diseases? They, would call them things like humors because they thought that it was just like some weird gas, some like almost spiritual gas that would make people sick. They really had no idea. There's all sorts of old stories about, we all got sick this summer because we didn't leave enough of our last harvest for the gods, so the gods are punishing us. Before we had this, we had no idea. So what really just blows my mind is that look at the date of Robert Koch. 1843 was his birth, 1910 was his death. So our great-grandparents, great probably, had no clue about what caused disease. No clue. 1843 to 1910. Like, I know my grandmother was born in 1903. So her parents were just on the cusp of starting to hear about, you know what, there's these little things inside our blood and maybe in the water and maybe transferred by mosquitoes. I mean, they had no clue. That was just the start of being able to understand that these tiny little things that we now know as bacteria, and they couldn't even see viruses, but these little things that we see as bacteria are what make us sick. Okay? And think about how much more you know. You know what a coronavirus looks like now. Okay? And you know a lot about how it's transferred. So much more than your great-great-grandparents would have known, because science hadn't figured it out yet. So, that's been another 100 years. If we look at that timeline here again, let's look at this timeline. So it was Robert Hooke in the late 1600s, and then Leeuwenhoek in just barely a little bit later than that, early 1700s. And then we have Pasteur in the late 1800s, and then Cook in the even later 1800s up until the early 1910s. Okay? So it's really a recent field, this microbiology field. And think of how far we've come in just a couple of 100 years. Okay, so that's really all of the history that we're going to do. I think it's fascinating. I highly recommend, if that piqued your interest, to find some little YouTube videos about those guys, because their research really is pretty interesting. Right. All right, so let's move on then to naming microbes. And this same thing goes, the same naming system works for all living species, plants and animals, us, fungi, everything, but not viruses. Okay, viruses are different because they're not living. So we use a process called binomial nomenclature. Binomial means two names. Okay, so we have that official scientific name. And so the example that they're giving us here is Escherichia coli, or E. coli, or sometimes you'll hear it pronounced E. coli. Okay, and that is the scientific name, Escherichia coli. And in this example, we also use that as the common name too. It's just E. coli is usually how you'll hear it. Okay, but with some other species, like I don't know, we use red oak. Red oak, the tree, is obviously the common name is red oak. The scientific name is Acer rubrum. Okay, so they're not always going to be the same scientific and common names. And the common names can be very different in different parts of the world. Like we might call it red oak here, and another part of the world might call it broadleaf oak. Think of something like, oh gosh, bird species. Like we have an American robin, the robin that we all know with the big red breast. But in Europe, they have a European robin that really doesn't look anything like our robin. But it's just, so we can have one species with multiple common names, or we can have two different species that have very similar common names, but the scientific name, the binomial nomenclature, is very specific, and each animal gets its own. Okay? Each bacteria gets its own. So there's only one Escherichia coli. Okay? There's only one species of Homo sapiens. That's us. Okay? So let's talk about how we write that out. Okay, as you can see here, this Escherichia coli is the proper way to write it if you are typing it. Okay? And that means italicized. Okay, if you're writing it out by hand, you should underline it, because you can't really write in italics unless you're very careful about how you write. Okay, so if you're writing it, you should underline it. If you're typing it, you should italicize it. But you should always put a capital at the beginning of the genus name, which is this first word. It's always going to be the first word is the genus name. And then a lowercase for the species name. So coli is the species name. Okay? And you can abbreviate these. You will find that they do that quite a bit in scientific literature, or if they're talking about something, multiple times. You put it in a news article one time, and you might call it Escherichia coli first, and then after that, you just call it E. coli. Okay. And when you do abbreviate it, this is the proper way. A capital letter to indicate the genus name, and then writing out the species name. Okay, so I want this also to be clear. Think back to our organizational scheme and how we had domain, kingdom, phylum, class, order, family, genus, species. So there can only be one Escherichia coli, but there can be many species within the Escherichia genus. Okay, so there might be 10 different species within the Escherichia genus. Okay. Also, to kind of confuse that a little bit, you could have, like I used the term red oak before, Acer rubrum. It's the rubrum part that is the red part. And so, of course, there's certainly another species that might be like Aristofolia rubrum. So something else can have the species name. But what is unique is this combination. Escherichia coli, there will never be another species that is named that. Acer rubrum, there will never be another species that is named that. When you put the genus and species names together, that is like its first and last name. Okay, just like ours. Except theirs is a little different, right? It would kind of be like last name and then first name, because we get less specific and then more specific. Okay, one last thing to mention is that with strains of bacteria or subspecies of, for example, fungus, or even birds even have subspecies, we will usually have some kind of identifier after the scientific name, okay, to identify specifically what strain or subspecies it is. For example, some birds, like the meadowlark, there can be an eastern species and a western species, subspecies, excuse me. So it might be like meadowlark eastern or meadowlark western, and then there's a caveat there that tells you more specifically what's what. Okay, so get used to writing those names out like that. You won't have to memorize any spellings of these, but if I ask you what is the proper way to write it out, you should say, I capitalize the first letter of genus, lowercase all the rest, underline it. And then if I had to type it, it would be italicized, again, capital letter first, everything else lowercase. All right. So another thing that we have to do with microbes is talk about how we measure them, because they are so very small. And it's kind of hard to even imagine how small they are when you start looking at the scale of these things. So if you think about a ruler and millimeters, like, if you stack, I don't know, 6 sheets of paper together, that's about a millimeter. And these microbes are measured on the micrometer or nanometer scale. So a micrometer is going to be it's going to be 10. You can fit 10 micrometers into 1 millimeter. That's what I'm trying to say. And the same thing for nanometers. It takes it down one more. So you can fit 10 nanometers into 1 micrometer. So we're talking extremely small. And this picture is good for showing the range. And I want you to spend quite a bit of time having this make sense to you. Okay, what I mean by that is it's a pretty important concept to understand what the size of all these microbes are. Okay, so let's look at this in depth a little bit. If you look down here at the bottom, it's telling you which ones are visible by which types of microscopes. So this red one right here is unaided eye. Like everything that's in this red band, you can see with the naked eye. So this right here is a liver fluke. It's like a flatworm creature. A yeast cell is here, and these are very hard to see with the naked eye. This is like tiny, but you can see them. If you had them in a liquid, you could see very, very small dots. You could tell there was something there. Same thing with this big algae. This big algae you can see with the naked eye. This is fungus, and this is a tapeworm. So you can see all of those with the unaided eye, okay? Now, if we go down a little bit, you see that this is the light microscope range, and it does overlap a little bit because the lowest level of light microscope would be like, 40 times magnification. So you can, still see that stuff, it's just bigger. So what can you see with a light microscope? Obviously the fungi, anything that you could see with the unaided eye, but then we're also adding the protists in. And to be honest, there's even a couple of protists that you can see with the naked eye, pretty darn close. And then bacteria, the very biggest bacteria, you can see with a light microscope. Red blood cells are teeny tiny. They're some of our smallest cells. You can see those. But anyway, so bacteria is going to take the light microscope. And then the electron microscope is what we're going to need to see the viruses. Okay, so now we're in this blue range, the electron microscope. And you can see it does overlap because it can see the big stuff too. But we don't see viruses with a light microscope. You can see that cutoff is right there for sure. Okay. All right. So before I go any further, I'm going to go back to share my screen, and there's a super cool, I don't know, little tool on the internet that was put out by the University of Utah. So let me go back here to share another screen. Okay. Hold on. Let me get pull this over. All right. Okay, so this is, like I said, the University of Utah, and it's looking at the size and scale of different common objects. And you'll see down here at the bottom, they tell you how many units of measure go into each of the other units of measure. And so as we go down, you'll be able to see that some of those flash by. Up here in the top left, this little grid right here is showing you the size of each of these. So right now, this little square is 1 millimeter. Okay, so that means that this little coffee bean is like 1, 2, 3, 4, 5, 6, 7, 8, maybe like 9 millimeters across. Okay, so that's what we're looking at here. You can see a grain of rice, a sesame seed, and as I move this little bar, we're going to get more and more focus. Okay, this right here is just a piece of paper on the bottom. Okay, so as we go more and more focused, you'll be able to see what happens to our millimeter right here in the upper left-hand corner. It's starting to get a little grid, and it's getting bigger. So we're focusing in, we're increasing our magnification of these objects. Okay, so I'm going to stop here because now we are just about to the next level of magnification. See here, this used to be our one tiny little millimeter, and now look, our 1 millimeter is much bigger. It's this big pink square, okay, which we can see is made up of 10 in one direction, 10 little, what's after the millimeter? See, this is the millimeter here, so we're going to go one step down into the micrometer range. So this little dot right here that's colored a little bit darker is going to be our micrometer. Okay, so we could still see this obviously with the unaided eye. This is a grain of salt. I'm going to go a little bit further. We can see an amoeba with the naked eye, and that's about as small as our naked eye is going to be able to go very well. You can see we can still see some of these other things down here, but they're becoming harder to see. So a good size amoeba, which is just like, it's a one-celled organism that kind of oozes around, and it does eat other, like bacteria, for example. But you can watch them crawl across a piece of glass. Okay, so anyway, this is about the range of the unaided eye. We can see that our millimeter is huge, and we are, definitely getting into the size of a micrometer now. And here goes our micrometer becoming bigger now. Okay, so a paramecium is a type of protist. That's one of our eukaryotic microbes. This human egg, this means like the ova that's inside of a female, and here's the tiny little sperm next to it. So that's pretty cool to see the size difference between those two. What's interesting, this photoreceptor is the rod, like the rods and cones that are in the back of your eye, and a skin cell. So you can see a human egg with the naked eye if you know exactly where to look for it on a slide. Okay, but you can't see the sperm with the naked eye. You can't see these skin cells and photoreceptors. All right, so this blue square up in the left now, this is our micrometer. Okay, so that micrometer, and then the next one down is going to be a nanometer. So this little box in the corner is a nanometer. Let's keep going. We'll go until the skin cell and the human egg are kind of out of the way, so we can see their size. And now look at the difference between the skin cell and the red blood cell that carries the oxygen. Okay, look, we've changed color. We're definitely in the nanometer range now. So this red blood cell is 8 micrometers. And let's see, we've got Baker's yeast. This was the one that was very close in size to what Pasteur could see through the microscope to be able to tell that the yeast, he didn't know it was yeast, but that it was in there, and that's what was helping the wine to work, to the grape juice to become into wine. And if we keep going, that chromosome is one of our chromosomes. That's a pretty good size. And now we're finally getting down into the bacteria. Okay. This is like 3 micrometers, 1 micrometer. Okay. Mitochondria, this is part of a structure inside of our cell that helps make energy. The lysosome is something else in our cells that help to digest things and move things around. But there's that bacteria, and we can keep going smaller now. So bacteria, we need the light microscope to see for sure. Keep going down. Keep going down. So we'll leave just that edge of that bacteria up there. And now we can finally see the viruses. And we are now in nanometer range on our big yellow square here. And this little one's going to be angstroms after that. Okay, so we now have the viruses. And I'm going to pull but look at the size of the viruses compared to the bacteria. Isn't that drastic at this stage? We'll zoom in a little bit more. So the measles virus is a pretty big virus compared to like this bacteria virus is a virus that infects a bacteria, so it makes sense for it to be smaller. Okay, keep on going. Hepatitis is a very small virus. Rhinovirus that causes a lot of common colds, very small. Keep on going, and now we're down into the molecule stage. Okay, so just getting so much smaller. These are just molecules. A ribosome, hemoglobin that's inside of our red blood cells, transfer RNA, antibodies. Oh, we can keep on going. Phospholipids that make up our cell membranes, and then some protein pieces and glucose, the sugar. Keep going down to water molecule, all the way down to the carbon atom. Okay, so it's just a super cool tool, so you can see how wide the range is between these microscopic things. We just think that they're all small and that they got to all be about the same size. But this really shows how very different all of these can be. So I'll post the link to this if you want to mess around with that. It's just pretty cool to look at. All right. All right, back to where we were. So that's one thing I don't like about these images, is that it kind of shows everything as the same size, and you don't really get an idea of the fact that this scale across the bottom is changing so much. This seems like, 0.1 nanometers, that's not that different than 10 millimeters. Oh, that goes all the way up to 10 meters, but here is 1 millimeter. So it doesn't sound that different, right? 0.1 nanometers up to 1 millimeter. But you saw on that scale how drastic that is. That can be a huge size difference. All right. So the light microscope, this is one thing that you should know. It can only magnify 1,000 times. So that means that when you're looking at something under a light microscope, it can only be up to 1,000 times bigger, which is quite a bit. Like I said, we can see bacteria, but it's not a great look at bacteria. You can tell what shape they are, and you can see them moving, but you can't tell any detail about them. They have 2 sets of lenses, the ocular lenses, which go up against your eyes, and then the objective lenses, which magnify even more. Okay, on the next screen, I'll kind of show you that. And then to do that highest magnification at 1,000 times, you have to use oil to do it. And I'll explain on the next slide where that goes. Okay, so this is a typical light microscope. This is almost identical to the ones that we use in the lab at LVC. And so these ones at the top are the ocular lenses. And they usually magnify 10 times. Some microscopes will magnify 15, but ours do 10. And then the objective lenses, you can see that there are multiples of those hanging down from the microscope. So most of them are going to be 4 times bigger, 10 times bigger, 100 times bigger. Or sorry, 40 times bigger and 100 times bigger. 4, 10, 40, and 100. Okay, and what you do, which we mentioned on the previous screen, is that you multiply the ocular lens magnification and the objective lens magnification to get the total magnification. Okay, that's this little point here. So if, for example, you were looking at something using the 40 objective lens, and you're always going to have the 10 on the ocular. So that would be 40 times 10, so the total magnification would be 100. Okay, I'll write that out. So if we use oculars, are almost always going to be 10. And then let's say, for example, let's do a different one. Let's use the objective lens that's 100. So 10 times 100 is 1,000. Okay, and that would in this case be the oil immersion lens. Okay, so going back to the microscope picture, when you're doing the oil immersion, this longest, zoom in a little bit more. Oh, it's gonna let me go. Okay. This longest lens right here, this is the one that you will use the oil with. And basically, when you put the oil on the slide, it connects the slide to that lens. It gets that close. It's not showing that close here. But they get so close that the lens is sitting in oil and the oil is sitting on the slide. And the reason that they do that is because it condenses the light and helps it really focus. So it gives you a much more clear image at that high magnification. Okay, the other things, the light source has to be really good for a microscope, and it points it right up through the bottom to illuminate your specimen from the underside. So these things are like 1 cell thick. When you're looking at a microscope slide, it's usually 1 cell thick so that the light can shine right through it and you can see some detail. Okay? All right, so we can see up to 1,000 magnification with that one. The electron microscope then is so much better. So instead of going up to 1,000 times bigger, we can go up to 20,000 times with one type and 200,000 times with another type. I can't even fathom like the scale difference between that. Okay, if you're looking through a light microscope and you can see a bacteria and it's going to look like a little tiny dot or squiggle moving around, when you look at it 20,000 times bigger, you can actually see the detail on the edges of the bacteria or the virus. That's how they know what coronaviruses look like, is because of these 2 microscopes. Okay, so what these 2 microscopes can do is use electrons. So rather than using light to see something, which is how we all see everything, electrons go into a tube and kind of get shot at the specimen. Okay, so let's say, for example, we have a bacteria sitting on a little slide. The electrons are going to be shot at that bacteria. So we can never do electron microscopy, at least not now, with anything that's living, because the electrons would kill it anyway. Okay, it just doesn't work. So we can only look at dead things with this. But the difference between the two, between the transmission and the scanning, is how the information is recorded, basically. So in both examples, electrons are shot at the bacteria. In transmission, the electrons are supposed to go through the object, and they're picked up, like, think about it, by receptors on the other side. Okay, so it's going to scatter the electrons just a little bit, and so you'll be able to see where the electrons have bounced off, but they're going through it. That's why it's called transmission. They're transmitting through the object. and being red on the other side, basically. So those ones are going to look like very flat pictures because that's really what they are. They're just being shot through, and they'll be scattered a little bit to give different light and dark areas. The scanning electron microscope shoots the electrons at the object, like the bacteria, but then those electrons kind of get shot back at the receptor. So it's like when you put your hand in one of those like metal, like all the little pins that are suspended, and you put your hand up in it, and it kind of gives you a three-dimensional image of your hand covered in pins. You know what I'm talking about? Those like little kid toys. That's like how this works, but with electrons. So it takes a reflection picture of what the bacteria looks like. So it's really good at seeing the three-dimensional view of an image. Okay, not so good at picking up quite as much detail. The transmission picks up more detail. That's why we can see it at a higher magnification. Okay. All right, this is a picture of what one of those machines looks like. The electron microscopes are huge, and they are almost always going to give a picture to a computer. It's not usually something that you're looking at through the eyepiece. You're trying to make pictures of it. Okay. So we're going to talk briefly about eukaryotes, prokaryotes, and viruses and how they're different. So stressing again that eukaryotes and prokaryotes are living organisms because they are composed of cells and they can perform metabolic actions. Okay, so an organism is composed of cells. Those are the eukaryotes and the prokaryotes. Cells are the basic form of life, and here are just some little numbers for how that works in real life things. The most simple life forms, like bacteria, are composed of just one cell. The amoeba that I showed you a picture of, the one-celled oozy creature, that's just one cell, even though we can see it with the naked eye. Fruit flies have about 6,000 cells. Not that many, considering, you could count to 6,000. But, they're so small. Can you imagine a fruit fly just having that many cells and yet having a digestive system and muscles to make its wings fly and all of that? But we, humans, as big as we are, have 30 trillion cells. And, of course, something like an elephant or a whale would have even more. 30 trillion, quite a few cells. Okay, so eukaryotes and prokaryotes are different in these very basic ways. Prokaryotes are the earliest cells, and pro means before, so like very early cells. They do not have a nucleus, and their DNA is in a loop. So rather than having like one long double helix, like we do, that typical picture of DNA that you've seen, they have one loop of DNA. Okay. Eukaryotes have a membrane-bound nucleus, which is where their DNA is. And so that means that the DNA is kept in this special little packaged area of the cell. And their organelles are like basically little organs. They are specialized structures within the cell that are also membrane-bound and do their own certain little functions. Okay? So prokaryotic, early, simple, no nucleus. Eukaryotic, they came later, they do have a nucleus, and all these special little, quote, little organs. All right, here's an example of a eukaryotic cell on the left. Much more going on here. It has a nucleus, it has little organelles, and then on the right is just a typical prokaryotic cell. And it's shown big here because this is probably a transmission electron microscope picture. But you can see it kind of looks pretty bland inside. That does not mean that bacteria can't do much. They can do an awful lot, but they don't have specialized, quote, little organs inside to do it with. Okay, a lot of it's just going to take place with enzymes that we'll learn about later. Okay, so which ones are the eukaryotic organisms? Protists and fungi, as far as the microbes that we'll be studying. Protists and fungi. And so remember when we talked about that term protozoa versus protist? So protist is a very general term that refers to all the little creatures that aren't fungi and aren't higher organisms. And so protozoa and algae are the two microbe examples of protists. Okay, so and remember there are other algae and I wish we didn't call them that, but like, for example, the cyanobacteria is considered an algae even though it's actually bacteria. So that gets confusing. So anyway, protists, like protozoa and the bigger algae, are eukaryotes. And then fungi are the other eukaryotes as far as the microbes go. Okay, so the protists are single-celled. That's what makes them kind of special. They share characteristics with plants and animals. So in other words, they're kind of animal-like because most of them can move, but they are kind of plant-like because they will often have a cell wall, and some of them can photosynthesize like plants do. Okay, fungi are molds, mushrooms, and yeasts. And it's really just the yeast and the molds that can possibly make us sick. And they have a weird lifestyle. They are composed of branching filaments, and that's most of what they are. When you see a mushroom growing out of the ground, or a mushroom that you would eat, that's actually the fruiting reproductive body of the fungi. Okay, most of their life is spent in these branching cellular filaments called hyphae. Okay, here's a picture of some of the eukaryotes. Yep, these are all eukaryotes. So this first one is, this top left one, is a little protist. This right one is an algae that's a protist. And then this bottom left one looks like a yeast. And the bottom right one is another fungus, and these are the reproductive spores of it. Okay, so those are all examples of eukaryotes. Even though they don't look like a whole lot, they are way more specialized internally than bacteria are. Okay, so the prokaryotes are the largest group of living organisms on Earth, and they first appeared 4 billion years ago. So again, they were the first ones. By the way, the eukaryotes arrived about 2 billion years ago. So they're both very old, but the prokaryotes are definitely older. And they live everywhere, right? We talked about them last time, living in the air and the water and the land and in ice and in our bodies. They're everywhere. Ubiquitous is that term, right? Living everywhere. The two types of them are bacteria and archaea. Okay, and as far as this class goes, we are not going to talk a whole lot about archaea because we don't interact with them a whole lot. So for right now, anyway, we're just going to say that both of these organisms, both of these huge groups, are unicellular organisms, which means they're just composed of one cell. And they all help to recycle and decompose material. But the archaea are really special because they tend to live in very rough places, like in deep sea thermal vents and in Yellowstone Springs and in places like that, like the Great Salt Lake, places that other things typically can't live. That's where the archaea really thrive because they can do so many weirdo things with their special chemical features. Here are a couple examples of bacteria. So this one on the left was taken with a scanning electron microscope, and the one on the right was taken with a light microscope. All right, now lastly, here are the viruses. They are, again, not living, so they are in their own little discussion here. So they are the most diverse and numerous entities on Earth, meaning that there are more types of viruses than there are types of bacteria. Okay, the bacteria would be number 2 probably. They are not cellular, so they do not have a nucleus, they don't have organelles, they don't do any metabolism, they don't reproduce, they don't breathe, they don't eat. They're crazy little things. So how do they continue to exist if they don't reproduce? Our cells reproduce them. Remember, I've mentioned that before, and once we get to the virus chapter, we'll go in more depth on that. But here's a little image that kind of shows that. Seems like my Zoom is broken now. Try this. There we go. Okay, so the viruses, this is showing what happens. So up in the top left picture here, This is a bacterial cell, for example, and this is a little virus that comes to land on it. And all that the cell actually says, oh, I should let that thing in. And so the virus, it didn't do metabolism, but just chemically it attaches to the cell, and then the DNA gets in. So that's the little thing right here. It's just the genetic material that came from the virus. And then our cell gets tricked into replicating all of those pieces. So it replicates the DNA, and it replicates the covering. You can see all those little finished viruses here. And then those viruses bust out when there are so many of them. They're not physically doing anything. They're not like chewing their way out or anything. But they bust out of the cell, and the release of the viruses is usually what makes us sick. It's actually killing that cell. Okay, that's what causes most of the symptoms of a viral disease, is when the viruses leave the cell. Any questions so far? Nobody's chatting or anything, so, if you ever have any questions about this, let me know. Okay, so viruses are only genetic material surrounded by a protein coat. That's it. So they're like a little strand of DNA or RNA, and it's wrapped up in a little layer of protein, and that's it. Some of them, like the coronavirus, has another envelope around that. So we call the first layer the coat, and the second layer the envelope if they have the envelope. Okay, coronavirus does. So it would look very similar to this one on the left with these little spikes sticking out. And this is a transmission electron microscope picture for both of these. See how it looks flat? It doesn't look as three-dimensional. But it can give us a lot of detail about the edges that we might not be able to tell by just taking a scanning type. All right, so I've alluded to this several times now, and we need to make sure that we focus again on this taxonomy, this of organizing life. So I do want you guys to memorize this list in order. So domain, kingdom, phylum, class, order, family, genus, species. So I recommend just saying it again and again until you kind of have a flow to it. That's how I do it. Domain, kingdom, phylum, class, order, family, genus, species. It'll eventually roll off your tongue. It just takes a while. But what some students do until they've learned that is come up with like a little mnemonic device to help you remember that, like Dear King Philip, and then I don't know, it keeps going. So come up with your own if you need that crutch for now, but I do want you to understand the concept of this. So basically what we're looking at is the way that we organize living things, okay? So there are only 3 domains, and they are bacteria, archaea, and eukarya. Okay, those are the only 3, and I do want you to know those 3. So all the bacteria are in the bacteria domain. All of archaea are in the archaea domain. So what's in the eukarya? So there are 5 kingdoms. Okay, so bacteria goes with bacteria, archaea goes with archaea. And then we go into protists, fungi, and animalia. And that's it. Oh, wait, hold on. Not protists. Oh, gosh, that was a bad mistake. I'll retype that. Got to get that right. Hang on a second. Okay, bacteria, archaea, and bacteria. Okay. And in the kingdom level, it's bacteria, archaea, plantae, that's what I was forgetting, fungi, and animalia. So where did the right there? Where did the protists go that I was going to put in their own category? They're part of animalia. Sorry about that. All right, so plants, fungus, and animals are all with eukarya. Okay, so all of those go together here. Okay, so bacteria to bacteria, archaea to archaea. Eukarya has plantae, fungi, and animalia. Okay, about that. All right, and then as we go down, we get more and more specific. Okay, so let's look at this. This is an example of some of those. All right, so this one is looking, see at the top here, domain, kingdom, phylum, class, order, family, genus, species, down the left-hand side. And then across the top, it's showing you that these three are the three domains, okay? And then here's our kingdoms within, and so they're only showing us the kingdoms within Eukarya, Plantae, Animalia, and Fungi. And then they're following down on, and they're showing you a few. So if we look down here, we're going to get down to coyote, domestic dog, human, and gorilla. So they're just following down that chain. So at the phylum level, this is everything that has a spinal cord, chordata. So some of the other things might be sponges. They're animals, but they don't have a sword. And then we go one step more down, and it's class, and that's mammalia. Some of the other classes would be things like reptiles or birds. Okay, and then we get down to the order. So everything in this order is a mammal, but now we're getting more specific. So we have the order of carnivore or primate. Okay, then we get down to the family level, more specific. So this first one is canine. Canidae is like a canine, like a dog. And then hominidae means like upright, hominid, big-brained animal. And then we get to genus, and that's the very second to last most specific. So canis, so you can see both of these are canis species. The coyote and the domestic dog are both canis within that genus, and then they get more specific on the species. So we have coyote here as Canis latrans, and domestic dog as Canis lupus. Okay, and then if we follow then, done with that trail, let's go back up to mammalia and then follow the other side. So we have primates for the order, hominidae for the family, and then in genus you can have either homo or gorilla. Okay, humans are Homo sapiens, and gorillas are gorilla. Some of them are redundant like that. Okay, so that just gives you a small piece of how these classification systems can work. So you can see we're more closely related to gorillas than we are to dogs because we branched off back here at the order level. So we are the same class as dogs. Of course, then that means we're the same phylum, kingdom, and domain as dogs. Okay, but once we get to the order, then we split off. So it just becomes kind of interesting. I don't know if you like taxonomy. I think it's fascinating to look at all these tree of life pictures. Okay, so this is a tree of life that's in your book. Very nice description because it shows the three domains. Okay, on the left-hand side here is bacteria. In the middle is archaea, and on the far right is eukarya, the one that contains the plants and fungi and animals. Okay. And so it shows you how they're related too. So there was some common ancestor that gave rise to both bacteria and archaea. And then they believe that this one's a little bit harder to pinpoint, but one of these archaea was probably a good developer and was able to just branch off huge into this whole field. Okay, so in other words, this does clearly show that we have more in common with archaea than we do with bacteria. Okay, there's some real fundamental differences to the cellular structure there. Okay, in the past, like a couple hundred years ago and more, they would put plants together that looked similar or animals together that looked similar. But now that we have all of these molecular tests, we can now classify things completely differently. And it's been fascinating to watch how many species have to shift around because we now have the ability to test their genetic makeup. Okay, so that's a huge evolving field. All these things are changing so quickly. All right, so anyway, this is just a cute little thing that's looking at all those different groups. As I was looking for tree of life pictures, I found that fun. That one's kind of cute. All right. So taxonomy is huge. I already mentioned this. It's, we're using DNA and genetic sequencing to really delve more deeply into this process. I'm not going to watch that video about medicine from fungus right now because we're kind of running short on time. I want to make sure I get through a few more of these. Okay. If we can catalog life correctly, then we can learn a whole bunch of new information from it. Okay. We have the ability to control invasive species, to develop antivenom and new medications, to understand if we, for example, have a turtle that's about to go extinct, could we use a different species of turtle to raise its young because they're so genetically similar? So there's so many new pathways within this field. It's really a growing field. If you want some to be guaranteed a job, become a taxonomist because there's still plenty there. If I come back, we'll watch this video about medicine for fungus. All right, so we're going to look at just a few of these tree of life images. This one's just a pretty one that just shows you in picture form the variety of living things on the planet. But what I thought was kind of funny about this one is that let's zoom way in, down here at the bottom, they barely even touch on the bacteria and the archaea. They're like these two little side leaves right here, even though most things on the planet that are living are bacteria and archaea. So this one really focuses just on all these living things up here that we can name most of. And here we are, just one of these little branches. This little one guy is the representation of Homo sapiens. This is a particularly interesting looking one. This is a tree of life that separates, see down here is the archaea. This one is made to focus on the archaea. So down here, this whole branch is the archaea. Sorry, let me put a rectangle around most of it. So most of this is archaea down here. Then this one over here, this is the eukaryotes. So this is all of us and all the mammals and all the birds and all the protists and all the fungi. All of that is there. In fact, this is us right here. We're in this epistheconts. We are just one little tiny branch. That includes the fungus. So in this example, we're thrown in with the fungi. And that means up here are all bacteria. And each of these little rays is like a whole class of bacteria. So what they're saying is that all of those little names that they've given are so distinct that they are more distinct individually, and they're worth mentioning individually, compared to our little branch of all the eukaryotes down here. There just aren't that many of us. We're really quite young if we're talking about the lifespan of the planet. Humans came very close to the end of it very recently. And this is another one that I think is just super cool. It's just another way of representing that same thing, that tree of life. And this is all bacteria over here on the left in black. And then the archaea here in this little brown area. And then this one focuses more on the eukaryotes because it's looking at the timeline of Earth, and this is the recent stuff. If you look at the scale over here, it takes a little bit more in-depth work to look at this one, but I just think they're all so pretty and they tell their own story. All right. Okay, so now I am going to go back and we'll watch that video. Let me see if I can get it loaded up here real quick. Might have to pull it out of my PowerPoint slide. So hang tight for just a minute. I'll have that video. Actually, no, I'm not, because I remember somebody saying that they can't hear the volume very well when I play a video through this. So I'll just make sure that, when you go to look at this PowerPoint, watch that video because it's really cool. It's talking about, this is Paul Stamets, and he's talking about all the different medications that we could find from fungus. And he's just like a mushroom guru who has devoted his life to mushrooms, and he has sent his mushroom extracts to this, I think it was the CDC. Oh, I can't remember now. Yeah, I think it was the CDC, Centers for Disease Control, because they're trying to find new ways to combat things like smallpox. So if somebody, smallpox is eradicated from the earth, but it still exists in the lab. If somebody got a hold of that and released it into the world, we have no immune resistance to smallpox anymore because we eradicated it, so we don't need to vaccinate for it anymore. So they had him testing some of his extracts for their ability to stop smallpox, and he found some. And so he has, contracts with the United States government to find new ways of pulling certain molecules out of mushrooms and using them to help find new medicines and new ways of stopping, for example, bioterrorism. So it's definitely worth watching. Check that out. Okay.